All right. It's 12:01, so I'm going to start. This is my pleasure to introduce Dr. Elena Choleris today, who's giving our Women's Health Seminar Series talk today. But before I do, I just want to acknowledge that and recognize that we live, work, and play, and we're so privileged to be able to do so and participate in a community on the unceded, ceded, and traditional territories of 203 First Nations, with 38 Métis chartered communities, each of which possess their own unique traditions and history here on this land in Canada, and what we now, or I now, refer to as British Columbia. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration of Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of our work, we are committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. And I encourage all of you, wherever you are in the world, to learn and understand the Indigenous territories of which you live, work, and play. So today, myself, I'm joining you from Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. We are the Women's Health Cluster. We have over 200 members across the world. Very happy to be able to say that. We run a number of events, of which one of these is the Women's Health Seminar Series. We're very fortunate to have picked International Women's Day to run 2 of our events today. We also have a brain health conference that we're running every month. It's all recorded, so you can watch these on YouTube later in your own spare time. We have a diverse set of researchers. We give out travel awards. Of course, nobody's traveling really right now, although hopefully we'll be able to do so soon. There are still costs with registrations and conferences and things. And so please, if you're a trainee, do apply for our awards and become a member. It's free and you get all sorts of wonderful things. And also I want to acknowledge our sponsors, because without them, we wouldn't be able to put on these events. UBC, Michael Smith Foundation for Health Research, and CIHR, as well as Elizabeth Rideout, who has donated a portion of her sex and gender chair in genetics to the cluster. Follow us today on Twitter if you'd like, Women's Health Seminar Series, that's what that stands for. Tag us if you will. And so I'm going to stop sharing my screen so that Elena, Dr. Choleris, can share her screen. And while she does that, I'm going to give you a very brief bio, and I'm going to try to, do a short fun fact, because Ellen and I have known each other for a number of years now, and have had many adventures when we could travel and go to conferences together. So I look forward to that again. But anyway, Dr. Choleris is a professor in the Department of Psychology and Neuroscience in the University of Guelph, which is in Ontario. She has a PhD in animal biology, which she got, and ethology as the subdiscipline at the University of Parma, which is known for its Parmesan cheese, right? I think that's the birthplace of Parmesan Reggiano, which I learned from Elena, among many things I've learned, not just about hormones, brain, and social cognition, but that Parmesan cheese is the true stuff, not the stuff that you find flakes in the grocery store. You should look for how many months old the Parmesan cheese is. There's a really lovely Stanley Tucci CNN. I don't know if you've been watching it, Elena. I think it's called Searching for Italy. It talks about food. But anyway, we're talking about Elena. Elena's been funded by NSERC for many years. She has won a number of awards, including the Discovery Accelerated Supplement from NSERC, which is a really prestigious award to win from NSERC. She won that in 2018. She has won a number of teaching awards as well at the University of Guelph, and she's a fellow at the International Behavioural Neuroscience Society as well. And she has a career. She was nominated for a Career Achievement Award also from IBNS. And she has over 100 publications since 1996, many of them in very high impact factor journals. You probably never heard of them, like PNAS and things of that nature. She is a fantastic researcher, a great speaker, obviously a wonderful teacher. She wins all these awards. And she has a really fantastic heritage. And this is my little fun fact about her. She didn't provide me with one, but I have many of them. But I'll just say that one of the times we met was at a conference in Torino, Italy, or Turin, Italy. And during a break, Elena disclosed to me she knew of this famous chocolate place, which is called Albiseren, and it was first the birth of Albiseren was 1763. So many years old. And she said, "Do you want to help me or come with me to try and find this amazing chocolate place?" And what do you think my answer to that was? Do you think I said no? Of course I said yes. And this is for the young ones in the audience. I know before you were born, it wasn't before you were born, but it was before cell phones could accurately locate you. So we actually really had some difficulty finding this amazing place. But ever since, we've gone back every 2 years to go to this fantastic chocolate place. Okay, so without further ado, you don't want to hear me talking about chocolate. You want to hear about Dr. Choleris, hormones, brain, and social cognition. So thank you, Elena.
Hello, everyone. So I'd like to begin today by thanking Lisa for a nice introduction and the invitation to talk to you. And also by wishing a happy International Women's Day to all women and to all men who care about women. And I hope all of you men who are connected right now belong to that group. I was going to look up why, and I forgot. So I don't know why, but in Italy, on International Women's Days on March 8th, women receive as a gift these flowers, which are from a tree called mimosa. So that is the symbol for March 8th. Yes, Lisa, is not only a cocktail made of champagne and orange juice, it's actually also the name of this tree, which flowers in the south of Italy at this time of the year. And yeah, so but I don't know why it came to be the symbol of International Women's Day for Italians. Anyhow, back to us. Today I'm going to tell you about hormones, brain, and social cognition. And before I even, okay, it's not advancing. Okay, I have to click on it to advance. That's fine. So before I forget to do that, I want to thank all of the fantastic graduate students who over the years in my lab have contributed to the data I will show to you today, from Anna Fan, who's now faculty at the University of Alberta, to some of the current graduate students, such as Dario Aspesi or Peter Paletta. Okay, so let's jump right into it. So in my lab, we are interested in social cognition. And as Lisa said, I am an animal biologist by training. And so I look out into the wild, into the real world. And when I see something like the variety that exists in the degree of sociality the species present with, then I wonder what is going on there, right? What the mechanisms of those differences are, because even if we just limit it to the mammalian species, we find that they range all the way from completely solitary to very highly gregarious and social. And of course, we belong to this group of social species. And so in biology, whenever we see something like this, we wonder why and we wonder how this is happening. And which is a short way of getting at Tim Berger's 4 basic questions in behavioral studies, where you will be wondering about the evolutionary adaptiveness of sociality, the behavioral mechanisms that allow social life, the ontogenesis, so the development within an individual's lifetime, as well as the mechanisms that underlie sociality, the neurobiology of it. And in my lab, we have been focusing on these neurobiological mechanisms of sociality for a number of years now, without ever forgetting the other 3 questions. So, and we have been looking at a number of systems that are involved in social behavior, including the sex hormones. And why the sex hormones? Well, because there are sex differences in pretty much every social behavior you look at. And that is true for Homo sapiens, for our own species, as well as most mammalian species. And this is a very incomplete list of all of the cognitive aspects of sociality that are that show to be different between males and females. And so in my lab, we have been then trying to understand how hormones modulate these social cognitive functions. The classes of hormones that we have been focusing the most are estrogens. We have recently started looking even at androgens, and right now with one of my graduate students, we are developing a study on progesterone. And today I will tell you mostly about the estrogen research that we have been doing, but also at the end, I will show to you some earlier results with the androgens. And we have been focusing on a number of brain regions. For estrogens, we have looked at the hippocampus, the medial amygdala, the paraventricular nucleus of the hypothalamus, and for androgens, the bed nucleus of the stria terminalis. And you will see as I go along why we have chosen those regions. So I will begin with the estrogen part. Estrogens are a class of hormones. The most common biologically are estrone, estradiol, and estriol. And I have highlighted estradiol here as the most abundant and biologically active in premenopausal individuals. And in terms of the source of these hormones, we all know the ovaries make them. And I hope as a group, you also know that it's not only the ovaries that make estrogens. A number of other organs actually have synthesizing cells, such as the adrenal glands, the liver, and I have highlighted here the brain, because in both males and females, turns out the brain makes lots of estrogens. And of course, for us neuroscientists, that is very cool and very interesting to then investigate. Estrogens act on cells in a few different ways. The better known and the first described process is the one often referred to as genomic mechanisms of estrogens, or classical, because again, they were described first. Through these pathways, estrogens enter cells. They do that easily. They are steroids, and as such, they are liposoluble. They easily cross cell membranes. They enter cells. They find their receptors within the cell, and then after binding to the receptor, they enter the nucleus of the cells and directly drive gene transcription. These effects, these genomic effects, are typically delayed in onset with respect to the application of estrogens, and it can take hours, even days, before you see the outcome of those genomic mechanisms, and then they also tend to last longer once they kick in. And then in the 2000s really became, even though it had been known for a while, in the 2000s, it became very much the focus of attention in the neurobiology world. The fact that estrogens also have very rapid effects on biological systems. And these very rapid effects kick in within minutes of estrogen applications. They act through a number of cell signaling cascades, and I will show to you results on a few of them today. And again, they act very, very rapidly on cells and organs. Estrogens act through 3 receptors: estrogen receptor alpha, estrogen receptor beta, and the G protein coupled estrogen receptor, which has been described most recently, named GPR1 or GPR1, because people, I think, expect that more will be discovered as we go along. So in my lab, we have been focusing over the past several years predominantly on the rapid short-term effects of estrogens and trying to elucidate the involvement of the 3 receptors, as well as some of the mechanisms underlying them. Today I will focus my talk on one cognitive process that is critical for social behavior, and that is that of social recognition. We study mice in my lab, and in a social recognition test, we determine whether or not a mouse remembers another mouse they met before by giving them a choice between a familiar mouse and a new unfamiliar mouse. We study social recognition because social recognition is the critical elemental cognitive skill that is required, is necessary in order for social interactions to be adaptive and appropriate. We all behave differently towards different individuals because we know who they are, and we adjust how we behave towards them using their knowledge and the past experiences that we have with that individual and all of that. And the critical cognitive skill necessary in order for that to be accomplished is being able to recognize individuals. And that is why social recognition is one of the key critical cognitive social skills that we have been investigating in my lab. Okay, so what do we know about estrogen and social recognition? Well, we know from some older gene knockout or gonadectomy studies that if you remove estrogens in the long term, you impair social recognition in males and in females. If you give estradiol, you can bring it back and you can restore social recognition. And the gene knockout mice have pointed at estrogen receptor alpha as being a critical receptor for social recognition, knocking it out, block social recognition. Estrogen receptor beta had mixed findings. So conservatively, those studies don't show a very strong involvement of ER beta in social recognition. And then, as I mentioned more recently, we have been started to look at the rapid effects of estrogens on social recognition. We have so far focused on females. We haven't done males yet, but we are starting. So we are catching up on the female bias that we have had in my lab over the years. And so with systemic treatments, we have found that shortly after administration, estradiol facilitates social recognition, and so do drugs that selectively target estrogen receptor alpha or the G protein capoestrogen receptor. The drugs instead that target estrogen receptor beta impair social recognition, and this is with systemic treatment in ovarectomized animals. So the next question after those initial investigations was where in the brain are these effects mediated? And I can tell you up front that the answer is not a simple one. It's not just one brain region. We are finding multiple brain regions, some that affect cognition in general, including social cognition, and others that instead seem to be more specific for social cognition. So where did we start? Well, we started by looking at where estrogens stick in the brain. This is a very old study that was actually published the year I was born, in 1968, showing where radioactive estradiol binds in the brain. And you can see a number of subcortical structures, mostly hypothalamic, but also some non-hypothalamic structures. And we actually decided to begin in the hippocampus. Even though estrogens don't bind strongly in the hippocampus, all three of the estrogen receptors are present there. And because the hippocampus is so involved in learning and memory, we decided that that was a logical place to begin. Certainly not to end there, though. Okay, so first the hippocampus. So we started cannulating mice, so implanting cannulas that targeted the dorsal hippocampus so that we could then later, after they recover from the surgery, we could infuse our treatments directly in the dorsal hippocampus. All of the mice in these studies were gonadectomized so that we could zoom in on the effects on the specific regions when we put in the hormones there. Okay, so then the first question was whether we were going to find any effects on social cognition and whether those effects were going to be specific for social cognition or could affect even other types of cognition. So we actually used a battery of tests. Social recognition is only one of them. We also do object recognition. We do the spatial version of object recognition, where it's the location of the object and matters. And we also do another kind of social cognitive task that I'm not going to tell you about today in detail, but I'm happy to answer any questions on it. And this is social learning, when animals transmit adaptive information to other animals, when animals learning from someone else rather than going through individual trial and error learning. So as I said, I will focus this talk on social recognition, but we did all of these tests. And in the first series of studies, we did administrations, either systemic or intrahippocampal, of either estradiol or the agonists, so drugs that selectively activate one of the three receptors, ER alpha, ER beta, or GPER. And then we look at what the effects were in those 4 cognitive learning tasks. And when administered systemically, estradiol enhances all of them. When administered in the hippocampus, it enhances all of them except social learning, which makes it interesting because social learning, as you can see, if you look at the first line here, tends to respond differently to this treatment versus the other learning tasks. So that makes it interesting for me. And then the other thing that is interesting in this table that is apparent is the fact that estrogen receptor beta seems to act on these tasks differently than GPR and estrogen receptor alpha. So while generally activation of ER alpha and GPR facilitates cognition, both when administration is systemic and when it is intrahippocampal, administration of estrogen receptor beta agonist can impair it in the case of social cognition or enhance it in the case of object placement, so the spatial task. And that is true both with intrahippocampal and systemic treatments. So once we found those effects, we started wondering about the mechanisms, and I will start showing to you actual data in a moment. In attempting to address this question, the first thing that we did is that we looked at dendritic spines, which are the sites of excitatory synapses on hippocampal neurons. So we did both systemic and intrahippocampal treatments, and we looked at what happens to the spines. And what we found, it's all published, so I just have a summary here added on this table, is that generally those treatments that enhance learning also enhance the number of dendritic spines in the dorsal hippocampus. Again, the exception is estrogen receptor beta, which either does nothing or actually has a small decreasing effect. Okay, so the next question we asked was, if those spines hold synapses, what is the nature of those synapses? And again, I have a summary slide because these are published data where a former PhD student, Anna Fan, did the electrophysiology studies and found that those new synapses induced by estrogens are immature and are actively kept silenced by the brain through a reduction in the glutamate AMPA currents, where the activation of AMPA receptors is the first critical step in learning. So what it looks like is happening is that estrogens provide the new spines, but they keep them silenced until an actual learning event happens. Okay, so that is our working hypothesis, that those spines are used when learning happens. And we have been trying to determine whether the hypothesis is correct or not over the past few years. We started by looking at the literature, and we look at the literature on spinogenesis, predominantly by Deepak Srivastava's group in London, England, who has been doing over the years some fantastic in vitro work looking at the induction of new dendritic spines by estrogens. And then we look at work from Karen Frick's lab. You should all be familiar with this name. I have put a smiley face there because she gave a talk for this group very recently. So you should be all very familiar with her work and even maybe this figure here, because she did show it during her talk. So without going into the details, what we did, we compare, right? We look at which mechanisms are critical for spinogenesis induced by estrogens and which mechanisms are downstream of estrogens activity in the dorsal hippocampus to induce the consolidation of memory when memory is tested long term. And we found a few that are shared. And so we started by looking at whether those same mechanisms are implicated in the formation of social recognition, so social memories by the hippocampus, and in a very short-term fashion. Mice, immediately after they learn, we don't wait for their memory to be consolidated. And so we decided to focus on ERK as a key player and on PI3K. We also did a few experiments on J and K, but I'm going to focus on PI3K and ERK for this talk. So this is how we test social recognition. As I told you, we give the mice a choice between a familiar and an unfamiliar mouse. So we initially habituate them 2 or 3 times to 2 mice in their cage. And then at test, we replace one of those for a new mouse. And as I mentioned, the test happens right after those initial habituation sessions. So it's very short-term memory that we study. All of our experimental mice have a brain cannula that allows us to infuse the treatment, in this case, in the dorsal hippocampus. And then we look at how much time the mice spend investigating those 2 mice that they receive in their cage. And we calculate an investigation ratio as our measure of social recognition. The investigation ratio is the amount of time the mice spend investigating a novel mouse over the amount of time they spend investigating both mice. And so a high investigation ratio is indicative of social recognition because it's indicative of a high level of investigation of the novel mouse. And that's what mice will normally do. Between something novel and something familiar, they will investigate the novel stimulus more than their familiar. And that's how we know that they recognize the familiar stimulus. In this case, the stimuli are all social. Okay, so this is a result of a study that has looked at the investigation of the involvement of ERK and the phosphorylation of ERK in the effects of estrogens on social recognition. So we have a task here in which with 3 habituation sessions, our control mice, which are all ovariectomized females, show social recognition. And we can tell that by looking at the investigation ratio. So when during the habituation sessions where the 2 mice are equally familiar or novel at the beginning, the investigation ratio is around 0.5. That is a random choice, 50%. They investigate both mice equally. When they remember the familiar mouse at test, we find a significant increase in the investigation ratio compared to habituation. So when you see stars up here, it means that there was a significant change from habituation to test for that group. And as you can see, when we prevent the phosphorylation of ERK, which is the activation of ERK, in a dose-dependent manner, we find an inhibition of social recognition. And the highest dose here blocked it altogether. And this is work by Paul Shepard, who was a former PhD student in my lab and actually also did a brief postdoc in Lisa's lab. So some of you may know him. Okay, so Paul found this ERK dependence of social recognition in dorsal hippocampus. And then what he did, he chose a dose that was not effective in it by itself at blocking social recognition. So this dose here, the 0.5 micrograms dose, because he wanted to see whether it could prevent the enhancing effects of estrogens with a dose that per se doesn't block social recognition. So that way he could see the actual interaction with estrogen effects. And so he did the critical study in which now he used a paradigm that is more difficult for the mice to perform, where our vehicle control does not show social recognition. There is no change from habituation to test here. And he replicated previous findings by Anna Fan, showing that infusion of estradiol in the dorsal hippocampus instead can facilitate social recognition. So it brings it back in mice that don't otherwise show it with this, as indicated by this significant increase here. And then the critical group is the one on the right here with the reddish bar, where he found that the sub-effective dose of the ERK phosphorylation inhibitor could prevent the enhancing, facilitating effect of estradiol. So that indicating that phosphorylation of ERK in the dorsal hippocampus is necessary for estradiol's facilitation of social recognition in a very rapid time frame. Next, Paul also looked at which of the receptors for estrogens mediate this effect. He didn't do ER beta because we had found no facilitation of estrogen recognition with an ER beta agonist in the dorsal hippocampus. He looked at ER alpha agonist, PPT, and G protein coupled estrogen receptor agonist, G1. And what he found is that while both PPT, so ER alpha activation, and GPR activation facilitates social recognition in the dorsal hippocampus, only for ER alpha, this effect was prevented by inhibition of ERK phosphorylation. For G protein coupled estrogen receptor, instead, in this group here, the ERK phosphorylation inhibitor did not prevent the enhancing effect of G1 of the agonist on social recognition, as indicated by the fact that this difference was still highly significant in this group. Next, he looked at the other of those two pathways that I pointed out at earlier, PI3K. And in this case, he found that PI3K is involved in estradiol enhancement of social recognition. And when he inhibited its activity, it could prevent the facilitating effect by estradiol. And when he looked at the specific estrogen receptors, he found that both the facilitating effects of ER alpha and GPR activations could be blocked by inhibition of the PI3K pathway. So in this case, then PI3K seems to be downstream of estradiol activations for both receptors, ER alpha and GPR, in the dorsal hippocampus. Another thing that Paul did, sorry, the image partly overlap on the text. So the other thing that he looked at was at whether other critical steps in the formation of new dendritic spines were also necessary for estrogen facilitation social recognition in the dorsal hippocampus. And these two steps are, one is the polymerization of actin. This is required for changes in the cytoskeleton that then leads to the formation of the spine. A spine is a protrusion in the cytoskeleton in the cell, and that requires changes in the cytoskeleton, and actin polymerization is critical towards that. And the other thing that Paul looked at is protein synthesis. So we know that the formation of new memories require protein synthesis. So Paul looked at the effects of an actin polymerization inhibitor and a protein synthesis inhibitor in his studies, and he found that both are necessary for estradiol induction of social recognition. So inhibiting those two processes with sub-effective doses that by themselves don't block social recognition was sufficient to prevent the facilitatory effect of estradiol. He also used a DNA transcription inhibitor, and that did not affect social recognition in the short term. And so that confirms that these rapid effects of estrogens are not dependent upon genomic activations, at least not in the very short time frame that we look at in our studies. So in summary, what Paul has found is that both PI3K and ERK are involved in mediating estradiol rapid facilitation of social recognition, and while for ER alpha both are involved, for GPR it's only PI3K as a pathways of those that we have looked at. What we still were wondering at this point is whether those new dendritic spines are necessary for social recognition, and also through which mechanisms, like which other pathways, or what is downstream of these specific neurochemicals. And there is still an open question that we will be following up in my lab next. Another thing that instead we have addressed at this point recently, we're still writing the papers. All of these data I'm showing to you today are new, non-published yet. So the other thing that we have shown is an involvement of the locally synthesized estrogens. So I told you earlier that the brain makes estrogens, and the hippocampus synthesizes estrogens. So we wonder whether these locally synthesized estrogens can be used by the brain to essentially regulate its own function in a very rapid dynamic manner that makes estrogens very similar to the action of a neurotransmitter in that regard. And so we look at the pathways of synthesis of estrogens. You don't need to worry about this really busy slide. The only thing that I want to point out here is how all of the estrogens are synthesized from androgens, and that is true both in the ovary and in the brain. And in the brain, there is a very high expression of the critical enzyme that performs this conversion. The name of the enzyme is aromatase, and there exist drugs that selectively inhibit its activity, and we use those drugs to investigate the role of these synthesized estrogen synthesized in the dorsal hippocampus on social recognition. This was work by Teresa Martin, a previous master student in my lab. She infused in the brain of ovary-ectomized mice, so there were no estrogens coming in from the ovaries in these mice. She infused an inhibitor of the aromatase enzyme, and what she found is that using a paradigm that is easy for ovary-ectomized mice to perform, where they show good social recognition, she found a block by letrozole infusions in the dorsal hippocampus. And she found actually the same effects for object recognition, less so for object placement, suggesting that these rapid enhancing actions of estrogens on cognition in the dorsal hippocampus can be mediated entirely by locally synthesized estrogens. Then another question that we have recently addressed was about the location of those receptors that mediate the very rapid actions on social recognition. I showed this slide to you earlier. I told you that estrogen receptors are found intracellularly. I didn't mention at that point that estrogen receptors are found even on the cell membrane. And it is believed that most of the very rapid effects are actually mediated by these membrane-located receptors, but there is some indication that some of those rapid cell signaling cascades can be also activated through intracellular receptors. So we wanted to know whether the membrane estrogen receptors are the ones that mediate these effects. All three of the estrogen receptors have been found on the cell membrane, so it could be any one of them. So we did a study in which we infused in the dorsal hippocampus estradiol conjugated to a very large protein, bovine serum albumin, which prevents estrogens from crossing the cell membrane, because this large protein is not lipid soluble, so doesn't cross cell membranes. And then we went to a very long, we put a lot of effort into ensuring that that's all we were infusing. So we purified the solution, we filtered it to remove any non-conjugated estrogens. We tested the presence of non-conjugated estrogens in the solutions, and it was zero, it was non-detectable by our assays. So we are confident that what we were infusing was only the BSA conjugated estradiol in this study. The study was performed by Talia Kuhn, who just recently defended her master thesis in my lab. And what she found is using the difficult paradigm where there is no social recognition of a control group, she found that infusion of the conjugated estradiol facilitated, restored social recognition in those mice. Thus demonstrating that its membrane estrogen receptors alone can mediate these rapid effects. And she found the same effects on object recognition and even the spatial version of the test. So it seems to be across cognitive skills in the dorsal hippocampus. Okay, so the summary so far in terms of our studies in the dorsal hippocampus is that estrogens here rapidly facilitate learning across an array of different kinds of learning. They also induce dendritic spines and immature synapses. It seems that the vast majority of these effects are mediated by estrogen receptor alpha and the G protein coupled estrogen receptors, while ER beta activation only facilitates spatial learning. And in terms of the mechanisms that we are starting to investigate, we are finding that these effects are mediated by membrane estrogen receptors. ERK phosphorylation and PI3K are both critical second messenger systems necessary for those effects, as well as acting polymerization and locally synthesized proteins and estrogens. So once we look at the dorsal hippocampus, we are still doing studies with the dorsal hippocampus, but we started going beyond there. And the reason is that we are really very interested in social cognition in my lab, and the so-called social brain has been identified as a network of regions that come online in the mediation of social cognition. So we started looking at other areas of the brain that are part of the social brain, where estrogens have their receptors and where they can affect cognition. So we began, actually, we didn't begin here, but I will tell you first about this study where we focus on the paraventricular nucleus of the hypothalamus. You may be familiar with it because this little nucleus at the base of the brain does lots of things and has many synthesizing neurons, including in terms of the interest of my lab, oxytocin neurons. Now, oxytocin is a neuropeptide that has been extensively involved in social cognitive processes, including social recognition, and that is under the control of estrogens. So we wonder whether by promoting the action of oxytocin neurons, estrogens could then promote social recognition. And this is our working model in which estrogens act on paraventricular nucleus neurons. Oxytocin is also made in another nucleus, the supraoptic nucleus, but in the mouse brain especially, the PVN is the key player in the production of oxytocin. So for now, so far, we have focused on the PVN. So the oxytocin neurons here produce oxytocin. We think that estrogen receptors can act on them. 2 of the 3 estrogen receptors are found in the PVN, ER beta, and the G protein coupled estrogen receptor. Those neurons project to a number of brain structures. Even they also project directly into the bloodstream where they release oxytocin for hormonal action. But in the brain, they project to a number of structures, including the medial amygdala. The medial amygdala is a critical region of the social brain. It has a high expression of oxytocin receptors, and we know that these are critical for social recognition. So we think that by boosting this oxytocin action here, estrogens can facilitate social recognition. The medial amygdala promotes social recognition by processing social cues. In the case of rodents, these are mostly olfactory in nature from the main and accessory olfactory bulbs. Okay, so Peter Paletta, current PhD student in my lab, has done this series of studies for his master and PhD. So first, he infused estradiol in the PVM, and he found an enhancement of social recognition in the difficult paradigm where our mice don't perform it normally. And then he also infused an oxytocin receptor antagonist in the medial amygdala, and showed that when we used an easy paradigm where control mice do well, it could block it with, it could block social recognition with the antagonist. And just like Paul did, then Peter chose a sub-effective dose of the antagonist, meaning a dose that per se does not block social recognition, to see whether it could prevent the enhancement of social recognition induced by estradiol when infused in the paraventricular nucleus of the hypothalamus. So here we are talking about two different brain regions and two systems, right? Quite a challenging experiment to perform because it involves the cannulation of mice in two regions. Okay, so this is the critical experiment where, using the difficult paradigm, he replicated the finding that infusing estradiol in the PVN enhances, facilitates social recognition, and then this enhancing effects of estradiol could be completely blocked by the sub-effective dose of the oxytocin receptor antagonist in the medial amygdala. Next, Peter looked at object recognition, and he found that the infusion of estradiol in the paraventricular nucleus does not enhance object recognition, and the blocking of the oxytocin receptor in the medial amygdala does not block social recognition, confirming what we thought, that the medial amygdala is a social cognition specific region where social recognition is processed. Okay, so back to the model. One thing that Peter still needed to demonstrate was that estrogens in the PVN actually do act on the oxytocin neurons, oxytocin system, and it did so by doing a microdialysis study. So in this case, he implanted microdialysis probes in the medial amygdala and the regular cannulas in the PVN, infused estradiol in the PVN, and then he collected sample of extracellular fluid in the medial amygdala, and then he measured oxytocin there. And what he found is that shortly after infusion of estradiol, there is a sharp increase in oxytocin in those samples collected from the extracellular fluid in the medial amygdala, suggesting that estradiol in the PVN can induce the release of oxytocin in the medial amygdala. The next, Peter looked at the role of the two of the estrogen receptors that are expressed in the PVN, estrogen receptor beta and GPR. And what he found is that infusion of the agonist for either of the two receptors could facilitate social recognition in mice. And then he did the critical study where he looked at the whether he could prevent those facilitating effects of estradiol in PVN by blocking the oxytocin receptors in the medial amygdala. And these are early findings. He just sent me these graphs this morning. And what he showed is that the oxytocin receptor antagonist was in the medial amygdala was able to block the enhancing effects of estradiol in the PVN on social recognition. Instead, for the estrogen receptor beta activation, we still are not sure because we have a trend here. So it looks like the oxytocin receptor antagonist is not blocking the action of estrogen receptor beta activation on social recognition. Peter just told me that he's still going through the placements of those cannulas in the brain. So these are not final results. So he's determining whether or not the cannulas were placed in the medial amygdala and PVN. And so these findings may change. They are preliminary. uh, I think I'm getting, I'm running late, so I think I will skip the next sets of results. All I'm telling you is that the medial amygdala also expresses estrogen receptors. And we found that infusing estradiol directly in the medial amygdala also facilitates social recognition. So the facilitation of social recognition can be mediated through action on the oxytocin neurons, but it can also be mediated directly by the medial amygdala. And we are still determining whether or not in here there is some interaction with the oxytocin receptors, or whether these effects are independent on those on the oxytocin system. So I'm going to skip these findings. And I want to just briefly mention some more recent results that we have in males. And for the male brain, we have started looking at the bed nucleus of the stria terminalis as a critical location for social recognition in males. This was shown with some older lesion studies. We wanted to determine whether this is also under the control of hormones in the male brain. And we began with testosterone. So this work by a current PhD student in my lab, Dario Aspesi, and what he's finding is two things. One is that social recognition is blocked in males by castration. So you can see here, after castration, males do not show social recognition in an easy paradigm where intact gonadal intact males show very strong social recognition. And then infusing testosterone in the bed nucleus of the stria terminalis could reinstate social recognition in these castrated males. So that was interesting in itself. And they also found that the same treatment in a similar time frame, so very rapidly, also increased dominance in these castrated mice, both in a very rapid time frame and even in a delayed fashion, even so most of his effects were much stronger in the rapid, through the rapid mechanism within the rapid time frame. And last, one thing that Dario is starting to elucidate is the mechanisms underlying those effects of testosterone. Testosterone is an androgen that acts on androgen receptors, but it can actually also be metabolized to two other hormones. One is through the aromatase enzyme I already told you about. The male brain contains lots of aromatase as well. So testosterone can be converted to estradiol and then act through estrogen receptor mediated mechanisms. Or through 5-alpha reductase, testosterone can be converted to dihydrotestosterone, which is a much more potent androgen compared to testosterone, has a much higher affinity for the androgen receptors, and does not, it cannot be acted upon by aromatase, so is not converted to estradiol. And in a first study, Dario has infused estradiol in the male brain. He still hasn't done the infusion of dihydrotestosterone, but what he has found with infusions of estradiol is a facilitating effect in the male brain. So this is males receiving estradiol in the bed nucleus of the stria terminalis, showing strong social recognition. So in conclusion, then, what I showed to you today is an array of studies where we have investigating hormone actions in the dorsal hippocampus, medial amygdala, paraventricular nucleus, the hypothalamus, bed nucleus of the stria terminalis, and how all of these actions through different mechanisms can all facilitate social cognition. And where we are heading is the identification of the network of brain regions that may be interconnected. We are looking a lot at this kind of interplay between regions recently, where hormones can promote social cognition, and hopefully we will be able to identify this network for males and for females. And eventually what we like to think is that our, which is very basic research done in a mouse brain, will be able to inform our knowledge of conditions in humans in which social cognition is impaired, such as autism spectrum disorder, social anxiety, schizophrenia. All of these disorders express differently in males and females in terms of their incidence, in terms of the symptoms that they manifest. And we believe that hormones play a major role in them, and understanding how that works will hopefully inform us on those conditions as well. Okay, last slide. I'd like to thank a number of collaborators who over the years have been involved in all of the studies I showed to you. I showed you the photos of my graduate students throughout, and so many undergraduate students that I cannot list them all here. But one of them is currently in Lisa's lab, Wansu, so hi Wansu, and she was involved in some of these studies as well. So I can, I guess, stop sharing my screen.
Yes, thank you. If everybody wants to join me in thanking Dr. Choleris for a beautiful talk. As always, so much data. We have a whole bunch of questions from Dr. Karen Frick, I can see, and Anna. I won't try to call. Also would like to ask a question too. So I guess Karen first, and then Anna. And I have a bunch of questions, but I don't know if we'll get to them. But
okay, hi. I will ask. Like, I'm not a neuroscientist.
Yes.
So can you tell in shortly from researcher, so what is exactly the gender differences for male and female brain? So because many people talk about it, but no one tells what's truth. Everybody has opinion.
Okay.
That's a very long.
Shortly, yeah.
It's a question that I can go on for hours, but the short is a number of sex or gender differences have been identified in the brain, and they go in different directions, and they are at different levels depending on the region. Sometimes the region is larger in brain, sometimes some regions are larger in males than females, and vice versa. The differences may be at the level of the structures of the cells, not just the size, at the level of the mechanisms that underline some of the effects of hormones on the brain, like Karen Frick's lab has been showing in recent years. So the short answer is there are differences, there are many differences, a mosaic of differences. It's not as simple as this is a pink brain, this is a blue brain, you know, but when you zoom in on different regions and different cognitive processes, then you find lots of differences.
But what does it mean for everyday life? Like, because we are talking so much about equality, and if you want...
In everyday life, there are many differences between male and females. Right? And in our behavior, in the way we think, in the way we approach life, they come from the brain. Right?
I really liked just, you know, Dr. Karen Frick has a fantastic review article called It Takes a Neural Village, and I know she has a bunch of questions for you, but, you know, basically she's showing that for object recognition, and you're showing that for social cognition. But the other thing, or social recognition, social learning, I know it's different, but also what I think is really important for people to understand is that the neural village does take a neural village, but the neural village could be completely different in males versus females. And I think that is, you know, important to point out. And I thought when you ended your talk talking about autism and the different manifestations, this could absolutely speak to some of that as well. Karen, did you have some questions?
Yeah. Well, great talk, Elena. Enjoyed it, and was relieved to see that you know, a lot of what you're seeing for social recognition, we've seen for object recognition. Like, it's nice to see that the mechanisms, the estrogen receptor mechanisms that you're seeing are similar, because it's reinforcing to make sure that, you know, what we found wasn't an anomaly in some way. So it's nice to see, for instance, that ERK is involved for, you know, ER alpha, but not GPER. So that's wonderful. You said you also have been looking at junk. Can you tell us a little bit? I mean, are you seeing...
So we are finding a GPR story there.
Ooh,
so
that's exciting. Okay.
It's so your work with object recognition and consolidated memories, it's confirming in social recognition and non-consolidated memories, suggests that these are really very early steps in that process of memory that already the brain can use early, right? Before the consolidation is processed, is completed, the brain is already benefiting from these effects in terms of performing in those tasks.
Right, because these were pre-training infusions, right?
Yeah, so we
Okay.
infuse right before they learn, right? And then
Yeah.
the test is right after their learning. So there is no demand on memory beyond a 5-minute
Yeah.
test, right?
I
And
mean, that's,
so we are finding this. And what I'm starting to think is that the hippocampus can do all of this. And the hippocampus is not specific for certain types, or memories. So really, estrogens there can really boost its actions on a number of memory types. But then when we
Yeah.
start zooming into other regions, we are finding more specificity in terms of objects. We have done a whole array of studies on the perirhinal cortex, where it's
Right.
object, while in the media we are finding social and non-object. So
right.
I think the network of regions, the hippocampus is the one that does them all, essentially.
Right.
And then some other may feed into it more specific information.
Well, I was interested to see, like, the hippocampus wasn't part of your oxytocin, like your PVN medial amygdala circuit diagram.
Right.
I think it fits in there.
Yeah, not yet. As in, we haven't done the study yet, but there are extensive reciprocal projections.
Okay.
So I think once we start looking, we will add that as a network, not as an independent region.
Yeah.
That's
Okay.
my guess, but we haven't looked, so we just don't know yet.
Yeah. Do you, and then I'll stop monopolizing the questions, but have you tried, or are you thinking about silencing, for instance, the projections from PVN to medial amygdala to look and see whether, you can still, whether those can, to try to get a sense of the directionality of those connections?
Yeah, so that would be one of the next steps in my next grant.
Okay.
To start doing that and look
Well,
at
yeah,
direction.
I'm happy to help you with that. If it's something, we're happy to answer questions, help you guys get started, because that's definitely your next steps.
Yeah.
Okay.
Absolutely.
Cool.
Well, mindful of time, so obviously if you have to leave, please do, don't, it's not going to offend us if you have to get off the call. But I have a lot of questions. I'm not going to ask any of them, but this is the part where it's not fun to not be traveling. I hear a lot of people talking about how we're going to have a lot of Zooms even when we can get back to traveling again, but this is the part I think that I really miss, is being able to chat afterwards about some of the nuances of some of the work. But I think it's really brilliant, and some beautiful illustrations of, I mean, you got towards the end, but the sex differences and how estrogens might be mediating some of these behaviors in males and females, but from a different brain regions and different networks, which I think is really, really interesting. Not that I don't think female-only studies are also really interesting, which is a big part of why I think this cluster is important too. I think I would just be remiss in I just wanted your thoughts about some of the long-term effects of some of our social / physical distancing that we've been undergoing the last year, since this is what you study, social memory, and not social isolation, I know, but social memory and social learning. But do you have any quick thoughts for us at the very end of our talk, and then we'll say goodbye?
That's $1 million questions. I think the social scientists are having a blast in that regard, because we are all part of a huge experiment worldwide where they actually are doing a lot of studies. In terms of my personal contribution, my guess is that we may very well realize that there are sex differences in the effects of this isolation, not just because women with small children are busier with the kids and are publishing less, which has already been shown, but also maybe in a more cognitive and then possibly even emotional level. Some of the sex differences in some of those disorders may then reflect in the way when men and women respond to this situation. That would be my guess, really. And in terms of specifically social memory, well, I think two things are happening. We are all interacting less, and maybe we may be losing some of our, skills in terms of that. But at the same time, I think we are all becoming fantastic at identifying facial expressions from faces with masks.
Yeah, that's true too. Yeah.
Under much more challenging conditions. Like, I don't know, I feel like maybe I'm just deluding myself, but I feel like I can tell if someone is smiling, by just looking at the eyes and the parts of the face that we can see. And there is actually a well-known sex difference that women are better than men generally at identifying facial expressions of emotions from just parts of the face. So maybe men are becoming better, they are refining their skill by
Learning.
practicing.
Yeah.
Yeah.
That's really interesting. I didn't know that part. I do remember from my early lessons of drama in drama school that they would tell us like act happy with your eyes or sad with your eyes. So I try to, even if I'm irritated in a store, I try to make my eyes not look too irritated. All right. Well, thank you. If you could just join me again in doing an emoji or unmuting and clapping. Thank you so much. Elena, that was a fantastic talk as always. So much data, and I'm really looking forward to hearing more of the story. And with that, I shall say goodbye to everyone. Thank you for joining us
Thank you all
on
for being here.
International Women's Day.